Well, hello to today's presentation on biology exams revision. We are going to look at the exams we took on December 2022 before we left for the exams break. We'll be looking at the questions and then the answers. These are three videos that I have on question 1, 2, and 3. So part 1 tackles question 1. Without much ado, let's just go right into it. Right, so the first question asks you to explain turgidity. So turgidity, it involves many plant cell. You need to specify that. Alright. The plant cell is placed or must be placed in hypotonic solution, high water potential environment, high water concentration, what have you. Okay. Then the next thing is that the plant cell being more hypertonic absorbs water by osmosis. Okay. That the plant cell swells or enlarges, the cell membrane pushes against the cell wall. The cell wall stretches but does not break. And you can go on and on saying that it becomes rigid, tough, and so on. All these explains a cell or a plant cell in a turgid situation. So any two out of these will give you 2 marks. Let's go to the next question, which that's II1A hemolysis. So hemolysis is about animal cells, right? Specifically, you need to say red blood cell. It's not good enough to say animal cell, no. Alright, it's not good enough. Okay, so you need to specify. Why? Because in this case you are talking about hemolysis, which means hemoglobin is involved, and hemoglobin is only found in red blood cells, right? Okay, the red blood cell is placed in hypotonic solution, high water potential, high water concentration, right? Then you have red blood cell absorbs water by osmosis, red blood cell swells and enlarges just like in a plant cell. The difference here is that what? The cell membrane will break and then the hemoglobin is released from the red blood cell. So if you are doing this in the lab with a beaker with red blood cells, initially you have a very if you do your centrifugation and you have your red blood cells, you place them in distilled water to create that hypotonic environment that you need. Then initially you have a transparent distilled water. Nothing happens. Then eventually the whole transparent environment situation changes, becomes red. That's because the red blood cells are bursting and hemoglobin is being released into the outside of the cell. So the transparency of the water is lost and becomes red and opaque. So any two for this gives you 2 marks. Good. Then we go to 1BI, which asks for you to explain certain stages of the bacteriophage reproduction. The first one was assembly, and it's just putting together the part of the replicated viral parts like the DNA, capsid, protein codes, and so on. So this is just 1 mark. And yeah, the diagram is not required. Alright, in fact, there was no space for the diagram anyway, so we didn't expect you to draw it, okay, to explain. Alright, the next stage is entry. The entry simply means the nucleic acid is injected, or released into the cytoplasm of the host cell. Okay, so it could be a DNA, it could be RNA, it doesn't matter. There's the release of the nucleic acid. So this also gives you 1 mark. Replication. The replication is the reproduction of the parts of the virus or the viron. So it involves the nucleic acid, the original nucleic acid, especially in the case of retroviruses where they have RNA. So they tend to now convert their RNA to DNA, use it to make their protein parts, the proteins, which are the parts of their bodies like the spikes, the capsid, the protein codes, and so on. Then in the building of the the phage or the virus within the host cell, then it forms whatever nucleic acid it came with. However, if it came with DNA, it will still produce the same DNA. It's only in the cases of the RNA where you have the RNA then converted to DNA, so that once it leaves the host cell into the other environment, it can then, the outside environment of the cell, it can affect other cells in its original form. Okay, so yeah, now that takes us to BI, where we are asked to state the differences between lytic and lysogenic phases of bacteriophage production. So lytic phase was on the left, lysogenic on the right. So the first one, lytic phase, it is the active stage of the virus. Lysogenic phase, it is the non-active or dormant stage. All right. You can also say that the lytic phase, because the active stage involves the manifestation of the symptoms of the disease of that virus. Right? If it's polio, you see the polio symptoms, and so on. If coronavirus, you see it, and so on. The lysogenic phase also, you do not see the symptoms of the disease. Okay, the virus, the virus activities do not lead to disease, so we do not see the symptoms of the virus. All right. So you can also say that DNA is used to replicate the virus during the lytic phase. So you are producing part of the virus, you are making the nucleic acid, you are making its protein coat, capsid, spikes, and so on. Whilst for the lysogenic phase is more concerned with the DNA merging with that of the host and subsequent formation of more DNA of the virus as the virus or the viral DNA replicates. It also produces the DNA of the virus. The lytic phase, again, it occurs rapidly, and the lysogenic phase is slow and takes a long time. Okay, so any two of these or any similar or related points you produce or you wrote down will give you 2 marks. Okay, then with CI we are asked to simply define osmosis, and again osmosis has to do with water molecules. It's not about substances, it's not about particles, it's not about just molecules. It has to do with specifically water. If you mention water without mentioning molecules is fine, but you won't want to see water molecules there. So they are moving from a high region of high concentration of the water to a region of low concentration of water, because it's passive. So, and therefore we need that concentration gradient to be there, and that can only be there where we have 2 regions of different concentrations of or volumes of water, isn't it? So that water can then move from the where they are more to where they are low. That's all we need, and to a semi-permeable membrane until you can establish some equilibrium where there is no net movement of water. It means that equal amount of water move into both environments. Especially if you are using a model to explain it. Okay, so that's okay for osmosis. Let's move on to II, which deals with the factors affecting osmosis. For 2 marks, you have to provide 2 factors. So any 2 here: concentration gradients, pressure, hypertonic solution, surface area, osmotic potential, temperature, concentration of solution, hypotonic solution. Any 2 of these for 2 marks. We were to state for D 2 ways in which osmosis is important to humans. Said that absorption water from the large intestine or undigested food into the bloodstream. Okay, even though we have water absorption occurring in stomach and other parts of the digestive tract, or the alimentary canal can mainly most of the water is absorbed from the food, and that's what creates the semi-solid situation in the case of the feces that we pass out of the anus. Then we also have reabsorption water in the nephron, the kidney, during urine formation. We will deal with more of these excretory system, the kidneys, later on in the later courses as you get to the second year, third year. Alright, so we'll leave it at that for now. Then reabsorption of tissue fluid at the venule end of the blood capillaries. Again, this is also something I will do under circulatory system. So just hold your horses for now. Alright, so any two of these for 2 marks. You are to also explain for DII importance of osmosis to plants. Consider it causes tertiary in plant cells, which strengthens the plant. Opening and closing of the stomata of the leaves of the plant controlled by guard cells, and this chiefly involve the guard cells, right? Being flaccid or becoming turgid. When they are flaccid, they close the stomata, and when they are turgid, it opens up the curl, then creates a pore, a hole through which gaseous exchanges, water vapor, i.e. transpiration, so on can occur. Also, absorption of water by root hair cells, movement of water from the xylem element to adjacent cells, or involve osmosis. So any two here give you 2 marks. You have to explain the biological processes involved in the preparation of Kobe, or salted or tilapia, or generally fish. It can be meat, it can be anything. This question was just to bring home our understanding of some of the home processes, things that we do not has been going for years. Our mothers have been doing it, but we do not know. But they didn't know. But then through this schooling and all that, we want to just bring your mind that hey, it was nothing more than application of diffusion, osmosis, and so on. Okay, right. So fish is soaked or sprinkled with salt. Okay, so and this creates a hypertonic region. So hypertonic region has low water concentration. Okay, so this is mainly around the fish or outside the cells of the fish. Therefore, we have a concentration gradient being formed, isn't it? We have the low water concentration outside the cells of the fish or outside the fish, then higher water concentration inside. So you have a water concentration gradient being set up, isn't it? And therefore, allows the water to move from a region of high water concentration to low water concentration outside the fish. And as we know, the preparation of Kobe is another way of preserving fish. Okay, and you know that water is a major contribution to spoilage. Okay, spoilage of food substances, organic substances, and therefore we want to eliminate the water. So this allows water to constantly move out of the fish, and thereby you can keep your or have your tilapia for a long time. The shelf life is increased. You can keep it, especially where there's a bumper harvest. Now this is for the water. You can also talk about conversely using salt. Okay, using salt to explain. Because you have high amount of salt outside the cell, the salt can also move into the cell. So as water move out, salt comes in, thereby creating a salt situation. And salt situation, high concentration of ions, make microorganisms also unfavorable to grow within the fish and spoil the fish, and so fungus and so on. So any two, and this is diffusion. Okay, and that one, and is diffusion, doesn't, because that one involves ions, so it's diffusion. So we have both osmosis and diffusion occurring or being applied here as the biological process. So any two will give you 2 marks. Okay, so that ends the question 1. I want to thank you all for watching. Do not forget to subscribe, like, and pass any comments, bad, good, complimentary, not complimentary. Hey, you're all for it. Thank you so much.